Write down the base sequence of anticodon. This, you see now, anticodon of triplet 1. If they say anticodon, what are they looking for? tRNA. They're saying that the following sequence represents, okay, base on molecule X. Which way is molecule X? Molecule X is messenger RNA, meaning that what is here is messenger RNA. So if they're asking us transfer RNA, then automatically we have to convert it. All right. So what is the next? The table below shows the amino acid that correspond with the DNA. You see, they are saying now corresponding with the DNA. So you have messenger, you have tRNA, you have DNA. What do they want? Okay, let's find out the question. What are they talking about in the question? Write down the correct sequence of amino acids. They are saying amino acid for triplet number 1 to number 3. For triplet number 1 to number 3. This is a different question. Write down the base sequence of anticodon of triplet 1. So it means that because this is messenger and this is transfer RNA, therefore I just need to convert this. Is A goes with U, G goes with the C, A goes with U. So that is the first answer. Then they're saying that write down the correct sequence of amino acid for triplet number 1 to 3, but they gave you DNA, meaning that what I'm going to do is, because this is messenger and then this is DNA, I have to convert the messenger into DNA. Because what is here is DNA and what is given is messenger. Therefore, I have to convert this messenger and then now I put it into DNA. So what am I going to do? I'm going to say it's going to be U, C, U, isn't it? Then I look for U. Sorry, for DNA, not you're converting into DNA. So it's gonna be T, C, T, because you're converting it from messenger RNA to DNA. So T, C, T is what? So is arginine. So the first amino acid I'm gonna write is arginine. Then the second amino acid is T. U goes with the A, and then G goes with the C. So it's gonna be TAC, which is methionine. Yes. And then automatically the last one, just to verify, it's gonna be CCT. So which is the CCT, which is the glycine. So basically, this is what you're supposed to write, but you write it in full so that you're able to get the answer. So you get it right. Yes, I wrote one. You wrote what? One. So you wrote them. So when it comes to meiosis, there are some things which you need to understand. Number one, what is meiosis for? What is it used for? Remember, meiosis is a type of cell division where one cell divides from 2 cells. After that, the 2 cells divide from 4 cells. And then this one comes as 23 pairs, meaning that 23 pairs of chromosome, meaning that it's 2n, which is equivalent to 46 chromosome. Then when meiosis 1 occurs, it's going to be 23 chromosomes, 23 chromosomes. They're no longer pairs. And then when this one split, then it's going to become single-stranded chromosomes, which is 23 here, 23 here, 23 here, 23 here. But what you need to know, meiosis 2, what you need to know that um, um, all of these are different from one another. They are different from one another. So it means that if this is a sperm, this is a sperm, this is a sperm, this is a sperm, all of them must be different sperms. That's why if different sperms they fertilize different ovums, though you end up producing non-identical twins. It's because of the different sperms which are fertilizing the ovum. So now what's the function of meiosis? Basically, the major function of meiosis is to produce haploid gametes. Number two is to produce what we call variation. Yeah, and not just variation, it's better to say genetic variation. Yes. Then another answer you can talk about is to maintain the chromosome number from generation to generation. Why like that? Is because during fertilization, the father brings 23 chromosomes, the mother brings 23 chromosomes. When they combine, you form 46. So meiosis is supposed to reduce them back to 23 so that the number of chromosomes in the individual is kept constant. So that is the meaning of that. So when you're done with that, this is what they are trying to say. Where do we find meiosis? So where do we find meiosis? We'll find out that meiosis, where do we find meiosis? We shall say that meiosis in male, you'll find it in the testis or testis, either way, because this is singular, this is plural. And then you also find it in the pollen, grains. Yeah, these are animals, these are plants. And then meiosis in female, you find it in the ovaries, in animals, and ovaries in plants. So if you look at the plants, the animals, they'll produce sperms. While plants, they'll produce pollen grains. Did I say it's found in pollen grains? No, it's found in anthers. Yes, sorry for that. It is found in anthers to produce pollen grains. So the sperms of the plants, they are pollen grains. So it results in the formation of pollen grains. While in animals, it is ovum. If there are many, we call them ova. And then in plants, it's ovules. So this is what we are trying to say here, that in animals, you'll find it in the ovary to produce ova. In plants, so in male, you'll find it in testes to produce spermatozoa, which are the sperm cells. And then in ovary, in plants, produce ovules, and then anthers to produce pollen grains. So that's what you need to know, where is it located. And then we said with the functions of meiosis, yes, we will see it when we look at this. Yes, we see that the function of meiosis at the end here, the importance of meiosis, that is to produce haploid sperm cells, that is haploid gametes. Those are the sperms and ova. And then number two, the halving effect overcomes the doubling effect. That's what I'm saying, that it maintains the chromosome number constant from generation to generation. So it has that. Then another one is to cause genetic variation. That is very, 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 very important. You have to know that it causes what? Genetic variation. This thing has refused. Okay. Then another one is haploid gametes, genetic variation, and then also maintaining the chromosome number from generation to generation. Those are the major functions of what? Of meiosis. So, what makes meiosis to be complicated? It's a simple thing. It's a simple what? Simple thing. This generation, this what? These phases. So it's simple. When you say metaphase, what you need to know is IPMAT, isn't it? IPMAT. Ne? Interphase, prophase, metaphase, anaphase, telophase. Interphase is what you call, I always call it growth phase. That's when it's growing. And then this is the reproductive cycle. So prophase is that we call the preparation. They are preparing themselves. Metaphase means they're in the middle. So if the chromosomes are like this, look, you must always look at it like this. When it's straight, ne? Because this is where the problem starts from. You see this? You see these are the centrioles, ne? And then these are the chromosomes. Yes, they're in the equator. Ne? So this thing must always be inside. Sometimes when we bring it like this. And then now when you get lost, where am I? Where is the equator? It's because now this thing is up, now this one up, this is down. Just turn your paper, ne? So that they face, these things must be on the sides. The centrioles must always be in the sides. So that's when you see that, oh, the chromosomes are in the middle. So, and then when you say anaphase, there must be a space in between. If it is chromatids, must have a space. If it is chromosomes, if they are chromosomes, you see, you can't say that this is what? Is metaphase. Why? Because you have a big space in here. So that space is what you call the anaphase, which means apart. That's what you call the anaphase, which means what? Apart. So once you know that, then at least you have identified the phases. Then telophase, automatically they have reached the terminal. But there are some few things which you need also to take note of this, that when it comes to meiosis 1, these chromosomes are always in pair. They are full chromosomes. Well, when you look at the same phase of anaphase, you find out that it's just these chromatids which are being pulled. You see, it's just, you see, half of a chromosome is being what? Not half. One side of a chromosome. You see, it's not full chromosome like this. So there's also space in between. But if you look at the chromosomes here, the chromosomes, they are full one. They have 2 strands. Well, here, it's only one strand being pulled. The same story here, the same story here. So you have to know, because this is where you start losing the answers there. So then another thing where you have issues, it's when we come to the karyotype. When we ask you questions concerning about karyotypes, and then you end up losing it. You don't know which one is a male, which one is a female, how many chromosomes. We're gonna reach there and then also see. So let's do some few questions about meiosis, and then we see we can also link it. Okay, they're saying that the diagram below shows the arrangement of two pairs of homologous chromosomes. Yes? Two pairs of homologous chromosomes in the cell. This is homologous to this. This is homologous to this. But now the question is asked, which phase is this? Anaphase, because there is a space in between. Centrioles are that side. So it means that it's anaphase, but because they are homologous chromosomes, therefore it's going to be anaphase 1, because we only find this in meiosis 1. So it's anaphase 1. Name two characteristics of homologous chromosomes. What can you say? They have the what? Same structure? Same height. Same size. They have the same size, the same shape, and the same genetic composition, or we can say that same
shape and size.
No, same size, same shape. That one is obvious. Then there is another one which we say that they have the same location of genes. Yes, meaning that if you have the location of gene for height or for skin color, in this chromosome, it must be in the same location as the other chromosome. So yeah, that's why I'm saying that same locus of genes, yes. So we have said that they have the same size. You understand? Same location of genes. That's why you see that. Now the white one, the white one is the same thing here. You understand? They are not the same. They are not the same. If they are the same, then we wouldn't have been different. But if crossing over takes place, then we would have been no genetic variation if they were exactly the same. What is inside them could be different, but the location of these genes is the same. So that's why I'm saying that same location of genes. So identify structure A. What is structure A?
What is A?
It's this spindle fibers. Spindle fibers. And they are saying that how many each of the following is present in the diagram. That's where the problem starts from. Yeah, that's where the problem starts from. Chromatids.
Yes, 4.
How many chromatids? How many each of the following? There are 4 chromatids.
Hey, how many?
Remember, the chromosome is formed by what? By 2 chromatids.
2.
So how many chromatids are shown? You have 1 chromatid, 2 chromatids, 3 chromatids, 4 chromatids, 5 chromatids, 6, 7, 8. So there are 8 chromatids because each chromosome, it is made up of 1 chromatid. Not 1 chromatid, 2 chromatids. Then they're saying that how many each of the following is present. The moment they start asking how many each of the following, now students will go off. Centromere. How many centromeres do we have?
12.
Because each chromosome is made up of 1 centromere. You understand? Yes. So each chromosome, 1 centromere. Each chromosome. So draw the diagram to show one of the cell that would be formed at the end of telophase 1.
One of the cell.
Yes, they're saying at the end of telophase 1. So I expect you to, this becomes a cell, and also this becomes a what? A cell.
So they are both cells.
They are going to be both cells, but they are saying telophase 1 at the end of telophase 1. So it means that what am I going to do is